This video will be the second part of Chapter 16, Control of Gene Expression in Bacteria. We'll look at several forms of post-transcriptional control in bacteria. Transcription occurs, you have the messenger RNA, and the regulation is really at the step of the RNA. So between this step, RNA to protein, there's some type of regulation. We're going to talk about three different examples of this. So antisense RNA, something called ribose switches, and also ribozymes. So this first example, antisense RNA, I'm going to tell you about this gene called the AMPF gene. So this encodes a protein that makes a channel for passive diffusion across the membrane. If the bacterium gets into an environment with very high osmolarity, it's going to actually suck all the water out of that bacterium, and you've really got to close off those passive diffusion channels very rapidly. So in order to do this, there's a couple of possible approaches. One way would just be to turn off that AMPF gene so it's not transcribed anymore. But that is not going to give a really fast response. So there's still a lot of existing RNA and protein that's in the cell, and that cell might die, in fact, before it gets rid of all of it. So what the cell does is it turns on this functional RNA gene. It's called MECF, and this gene is actually complementary to the messenger RNA for the AMPF gene, called an antisense RNA. And it binds to that messenger RNA and it prevents it from being translated. Ribose switches are another kind of regulation of RNA. So this is actually done by a regulatory protein. So this protein will bind to a messenger RNA and make that messenger RNA form into a particular secondary structure. When the structure is present, the ribosome binding site is actually hidden. So therefore that messenger RNA will not get bound by ribosomes and the gene will not be translated. In the absence of that regulatory protein, that messenger RNA forms a different secondary structure. Then that ribosome binding site, the Shine-Dalgarno sequence, would be accessible, and the ribosome can bind on there and translate the protein. The last example is something called a ribozyme. So a ribozyme is actually a RNA that has catalytic activity on its own. And what happens in this particular gene is there are two secondary structures that the messenger RNA could take, and one of those causes it to actually cleave itself and break itself down. So this particular gene encodes this RNA, you make this protein, glutamine 6-phosphate amidinotransferase, and the product of that protein is actually a molecule shown in blue here. And this product will bind to the messenger RNA that encodes that enzyme, and when that particular effector is present, that changes the conformation of the RNA and the RNA degrades itself.